All right, well, we'll go ahead and get started. Good evening. This is Susan Hedstrom, Executive Director of the Foundation for Prader-Willi Research, and I thank you for joining us tonight as we announce the results of our most recent grant cycle. 10 outstanding projects have been selected for immediate funding, and we have you, our families, hosts, fundraisers, event hosts, to thank for making this possible. It's your support that has allowed FPWR to fund PWS research and at an ever-accelerating pace. We are truly humbly grateful for that. Tonight's call should run for about an hour, and as always, we will save time at the end to address your questions. If at any time you do have a question, please use the GoToWebinar control panel to type and submit your question, and we will do our best at the end of the call to review all questions that have been sent to us. Without further delay, I would like to introduce Teresa Strong, our Director of Research Programs, and Jessica Bohunowicz, our Associate Director, to share with you the details on these exciting projects.
Thank you, Susan. This is Teresa, and I think I'm going to take the lead on the presentation, although I'm sure, or I hope Jessica will jump in and correct me if I say anything wrong. So thank you all for coming this evening. I know you all have busy lives, and I appreciate you taking the time to be with us and hear about our latest grants that we're funding here at FPWR. So I always like to start by reviewing our mission, because this is really our reason for being. It is what we focus on when we make all our decisions as an organization. So our mission is to eliminate the challenges of Prader-Willi syndrome through the advancement of research. And of course we do it for our loved ones and all of these beautiful kids that you can see down here, for our own kids, as well as the children of what are now our dear friends, who we never thought that we would necessarily become friends with, but we're all joined by this common goal. So I wanted to start, as I was putting this talk together this evening, I was reflecting on the fact we're starting into our second decade at FPWR. We were founded in 2003, and so we're starting our second decade of existence. And I was so thankful, reflecting back on it, for the support of our community. So for everyone who has run a marathon or hosted a One Small Step walk, or donated their birthday funds to us, we are so appreciative of that. And our increase in funding has really allowed us to do so much. So it's really encouraging to see how we're now starting to see the fruits of the seeds that we planted a few years ago. Science sometimes takes a couple of years for you to see the results of our studies, and it's very nice that we're starting to be able to see the advances that this funding has made. As well, since we've, especially in the last few years, we've really gained some momentum with our funding. It's really allowed us to have more impact on the field and really be targeting specific areas that are really important and bringing those along. So it's very nice to have the flexibility for me personally that the increased funding brings, because it allows us to really target things and advance them. And I just wanted to mention too that we're very cognizant of the fact that this money is not easy to raise. People are entrusting us with it, and we do try to take a lot of care in keeping our expenses to a minimum and funneling as much of the money as we can straight into the researchers' hands so that they can move things along. So there are two major ways that we advance our mission through our general grants program, and that's what we're going to talk about tonight, and that's what we call investigator-initiated research, which I'll talk about. We also, as I said, now that we've got a little bit more funding and we have the opportunity to do more planning and more I don't want to say thoughtful, because I think we've always been thoughtful with our funds, but longer term planning. We also have special research initiatives that are listed here that we can, that we've discussed some in the past and we will discuss in other webinars. But today we're going to talk mostly about our general grants program, which is the investigator initiated program. So the goals of our grant program are to support research that's going to significantly advance our understanding of PWS and or develop new therapeutic interventions for PWS. So we really want things that move the field forward. We're specifically looking for innovative early stage, high risk, high reward research. So things that may not, early stage things that might not have any other funding source to get them off the ground and get the researchers started on something new and interesting. Another goal is to expand the base of PWS researchers. We want to draw in scientists who have expertise in different fields that are relevant to PWS, but who don't necessarily do PWS research all day or as part of their career to date. We also are cognizant of the fact that we need to be bringing young investigators into the field so that they can establish their careers in PWS. And it's quite true that oftentimes the first couple of grants that an investigator gets, that's the field they're going to stay in for years. So we want to kind of hit them at that point. And finally, an important goal of ours is to involve parents in the process of identifying relevant research areas to pursue in research, and also in selecting the most deserving research protocols. So we, of course, want the science to be really good, but we want the parents to think to know that it's relevant to what's important in their life. And our overall objective is to establish a consistent source of funding that researchers can come to rely upon through a competitive grant process to foster research. For our investigator-initiated program, really what it is we ask the investigators to send us their best ideas. So we don't specify in this case. In this situation, we know that the scientists are thinking about PWS or their particular area of expertise all the time, and they're in the best position sometimes to identify really important avenues that can advance the research and the therapeutics. So we solicit it from scientists across a broad range of disciplines that are relevant to PWS, and then we review those projects both on the basis of scientific merit, and we get the scientists in the field to review it on that perspective, and we also ask our parent advocate reviewers to review it for relevance to our community. So to date, I'm just going to go through a few statistics about what we funded to date. With these new projects, we've funded more than 80 projects in total, with more than $4 million of funding over since 2003. The overall success rate for projects is about 27%. So somewhere between 25 and 30% of projects that are submitted get funding from FPWR. Here's some statistics that I kind of keep some running statistics on who is getting our funding. So to date, we've funded 58 different scientists, so 58 different principal investigators. And we do keep track of whether they're junior investigators, so early in their career, or senior investigators. And I think we want a pretty close to a 50-50 split of that, which is about what we have. So the senior investigators are great because they know the field, they've been in the field, they know how to get things done, and we want to be supporting them. They're kind of tried and true. We know they're going to be productive. The junior investigators, as I said, we want to be helping people start their careers out and hopefully staying in PWS research for the long term. So we want to fund both of those. We also we're a rare disease, and since we are a rare disease, we need to look all over the world for talented investigators who are studying PWS. And so we fund research. So far, we funded research in 11 different countries. We've received grant submissions from 14 different countries. The majority, about 65, 70%, are in the U.S., but as I said, in many other countries as well. And that's great, too, because especially as we move into clinical trials and the registry and things like that, we really want to have a global reach and a global community to look at these things. Finally, on the bottom left, you can see the split between researchers who I call PWS researchers. So those are investigators who have published in the area of Prader-Willi syndrome before. So these are people who are in the field, and non-PWS researchers shown in red. And those are investigators who have expertise in other fields, and this is kind of their first time looking at PWS. So again, the majority of what we fund is people who have a record of studying PWS, but we also want to be drawing new investigators into the field, and I think that's a really important mission for us to do. So we, as I said, our one major goal is to expand the base of researchers who are working on PWS by supporting young investigators, and here's some of the young investigators that we've supported, by recruiting some well-established researchers who have expertise in other fields into PWS. And so, for example, one of our major efforts over the last few years has been to get some really good neurobiology people studying PWS, and we've had some really good success with that, some of which I'll tell you about tonight. And then our other sort of target audience is established PWS researchers who are starting a new area of research, so pursuing some new directions. This graph shows the research categories. Our studies are based in animal models, so for example, the PWS mouse, or based in cellular models, and that's shown in blue, or clinical research, which is research that uses human subjects. And it's been interesting over the last 10 years, this clinical research has really started to increase, and I think that reflects some of the maturity of the field, as well as the maturity of FPWR, being able to support more studies that are directly impacting patients, and so more clinical in nature. And this graph on the bottom shows you the research areas which are really as diverse as PWS itself. So we have funded a lot of work, of course, in appetite and obesity research, behavior and cognition, neurobiology, endocrine disorders, and genetics. So there's a lot of things, a lot of areas that are relevant to PWS that might have an impact on our kids, and we've really kind of tried to hit a little bit of everything. So I don't have too much time tonight to go through our accomplishments to date. I just wanted to kind of highlight a few major points and just give you one example. So I mean, it's great that we've funded $4 million worth of research, but the question that all of you, I'm sure, have is, well, what does that do? What has that done for our kids? One way we can measure that is how many publications have been supported by FPWR funds. So the publications are the scientists reporting the results to other scientists so that other people can build on their findings. And to date, there's more than 50 publications in the medical literature that have been supported by FPWR. Another measure of how important our research is how our investigators do on a national, competing for additional funds. So what we want to do is give people a relatively small amount of money so that they can generate data that will allow them to get larger grants from the National Institutes of Health so that they can study something more in depth. So to date, our FPWR funded investigators have received more than $5 million in additional support from the National Institutes of Health or other government agencies. And that's to continue the studies that they've started under FPWR. So they get a year or maybe 2 years of support from us, they get the study started, and then they're able to secure a much larger grant on the order of $1 million or $2 million to do 4 or 5 years additional work and really build their program of research. We've talked about bringing new scientists in the field. Another thing that we've been able to do is help scientists develop some new resources that they can share with others, and that's a really important role for us, I think, as a patient advocate organization, because many granting agencies don't set aside a lot of money for that kind of thing, and it can really help spark a lot of research. And we've also really focused, again, what I won't talk too much today about is promoting interactions and collaborations among the scientists, and then collaborating with the National Institutes of Health, with the pharmaceutical industries, and also with the FDA to advance PWS therapeutics. So this is just one example of the returns on investments, and it's in teeny tiny letters. These are the publications from last year that were supported by FPWR funds, and you can see that there's just a lot of different topics, a lot of different areas, and I think, as I said, as we've been sort of building our research portfolio, we're starting to really see the fruits of that now with many aspects of both basic science and clinical science being published and shared among the scientists. And I'll just mention in a little bit more detail one of these projects, just to show you how these things evolve, and that is a project that was started actually as part of the, for those of you who have been around a couple of years, in 2009 we held an NIH-funded Prader-Willi syndrome workshop in which we brought together about 70 scientists and prioritized what areas of research we needed to be funding and to be supporting. So one of the things that came out of that was that we really needed to develop some of these, what was at the time very new, pluripotent stem cells from patients with PWS. So this is an approach where you can take the skin cells or blood cells of someone with PWS, you put some genes into them and convert them to where now you can make neurons in a dish. So basically gives you a way to study the brain of someone with PWS without having to sample their brain, which is a very nice thing. So we funded in 2010 and 2011 Mark Laland's group to develop some iPS cells, some pluripotent stem cells from individuals with PWS, and they did that, and they published on those cells and the characterization of those cells, and this is a picture of iPS cells. And then in 2013, we funded Krista Martins-Taylor, who had been a postdoc with Dr. Laland and had moved on to a faculty position at University of Connecticut. And so she was using these cells that he had developed to really understand how the PWS region of that chromosome 15 is regulated. And what they've just published, actually last week, so Jessica hasn't gotten a chance to blog about it yet, is the finding that that they can actually, they dissected out what proteins are regulating that region, and they show that they can reactivate expression of the maternal allele. So that is not something that would be translated into a therapeutic right away, because it's probably a fairly non-specific, it's not specific enough to be administered to humans, but it does show that it can be done, and that's one general approach to doing it. So that just shows you how these things take a couple of years to evolve and, you know, really see the outcomes that we want to see. Okay, so with that background, I'll launch into the new projects that we funded this year. So as I said, these are on all topics. The investigators have to show how it's relevant to PWS. And this is the grant cycle that we received the grants in mid-December 2013, and then reviewed them in kind of January and February, and made decisions earlier, well, last month at the end of March. We received 44 grants in response to this request for applications, which is the most grants that we've received to date. So the word is certainly getting out, which is great to submit grants to us. We had set aside $750,000 to support this grant cycle. And as you see, we spent a little bit more than that, but I think it's well worth it. So we've chosen 11 grants in total, which will add up eventually to a little bit more than $800,000. Those are shown here, and I'm going to break them down and kind of give you the highlights of those. Just a reminder as we go forward, I'm sorry, I'm a geneticist, I have to throw the genetic slide in there. This is just to remind you that individuals with PWS get it in one of three ways. They might have a deletion on the chromosome 15 that they inherited from dad. They might have two chromosome 15s that they inherited from mom and none from dad, or they might have an imprinting defect, which is a chromosome inherited from dad that actually behaves more like it's inherited from mom. The trick is in all of these cases the actual genes that we're interested in are on the maternal chromosome. They're all sitting there on the maternal chromosome. They're just silent. So when we talk about reactivation, that's what we're talking about, is trying to get the genes on the maternal chromosome activated. And this just shows you a picture of that region to remind you that it's a very complex region. We've got lots of different genes, and part of our problem in PWS is we don't really understand the contribution of each of these genes to the overall characteristics of PWS. We do know two genes that seem particularly important are the MAGL2 gene that will come up a lot this evening, and then the SNORD116 set of genes. It's actually a little cluster of genes. So sorry, those of you who don't like genetics, I'll be done with that now. We have some key questions, of course, that are important in our kids' lives. And one of the first ones is, why are people with PWS hungry all the time, and what can be done to change that? So our kids, when they're born, they don't, at least to the extent that we can tell, they don't seem to be really, really hungry. And then as they get older, at some point they start to develop hyperphagia to some degree or another. And it's variable between individuals, but that general pattern is pretty consistent in PWS. So we've funded 3 studies that will look at that question in different ways. And so let me just take you through those. So the first study is by Garrett Stuber at University of North Carolina, and his group has shown recently that if they enhance the inhibitory circuitry in the brain, and a particular part of the brain, that they can make otherwise normal mice eat excessively. So they're really getting at this mechanism of what's the circuits that make people eat. And they think that loss of the PWS region genes produce a similar effect. So they're going to look at this MAGL2 mouse, so the PWS mouse that's missing one of the genes in the PWS region, and that's a mouse that does get obese, and they're going to define the neurons and the excitability of those neurons and see how they compare to normal. So we're excited about this grant because this group is on, super cutting edge. They just had this paper in Science, which is a very big journal. Everybody wants their paper in Science last year, identifying this particular circuitry that is really important in feeding. And so to have this group really turn around and look at PWS, I think is very exciting. He's a very productive young neurobiologist. As I said, one of the things we emphasized after the workshop was that we need to bring new neurobiologists into the field of PWS. The long-term potential contribution is that if we precisely define the neurocircuits that aren't working in PWS, then that may direct us the development of new avenues of therapy. The second grant in this group is from Sebastien Bourdais, who's at University of South Carolina. This is the second year of his study, and he has shown that, again, using the MAGL2 mouse, that there's specific neurons called the POMC neurons that during development don't seem to be doing well. So his interest is looking at neuronal health and circuitry over time. So he is looking at sort of that period as animals grow from just being born into adulthood, and so he is based on what he's seen so far, he thinks that the cells in the PWS brain may be undergoing a higher degree of cellular stress during development. And so he wants to investigate that further. The other thing that he wants to do is see whether he can rescue that. So there's ways to rescue cellular stress to give the mice compounds and see if he can recover and maintain function of keep the neurons healthy that are otherwise having difficulty maintaining their neural circuits. So we're excited about this grant because it's exploring a whole new mechanism for what's going on during neurodevelopment in PWS. It uses the MAGL2 mouse model, which is a good model of PWS. And this lab is really dedicated to understanding how the hypothalamus development impacts appetite. And that's really important for understanding that switch when our kids go from not being so hungry to really becoming hungry. And it's also nice that long term there's the potential for a new therapeutic avenue to look at these ER stress inhibitors to keep the neurons healthy in the brain. The final one in this group of understanding hunger in PWS comes from Dr. Piomelli at UC Irvine, and he is looking at this compound, oleoethanolamide, which I will now call OEO, because that's much easier. And that's a molecule that's produced in the gut, and it actually is important in satiety, so making you feel full. And he is really kind of the world's expert in this particular pathway of, this molecule making people feel full. And he is hypothesizing that this pathway is disrupted in PWS. And so he has joined with Dr. Virginia Kimonis, who's also at UCI and who sees PWS patients. And he is going to look at whether the system is impaired in the PWS mouse. If it is impaired, he wants to treat those mice and see if he can, and they're using the SNORD 116 PWS mouse that is hyperphagic, so it overeats. So he wants to see if he can use this compound to make them more full and make them eat less. And they're also going to look at PWS humans, kids, and adults, and look at their blood levels and see if there's a deficiency of this compound in their blood. So this is interesting because it's potentially a new piece in the PWS puzzle. It includes both the animal and the human studies. This investigator is an expert in his field, and he's collaborating with someone who is an expert in PWS, so that's great. In the end, it may offer some new avenues for therapeutics development, which of course is which is what we want. So in addition to hunger and obesity, one of the major things that impacts our kids and our families is behavior and mental health in PWS. So another sort of group of grants that we have addresses the question of, can behavior and mental health problems in PWS be reduced or prevented, even better, prevented? And these studies are looking at autistic behavior, temper tantrums, and then mental illness. So there's a series of 4 studies that I'll go through that are addressing this question. The first study comes from Christian Schaaf, who is at Baylor College of Medicine. And Dr. Schaaf, he published a paper late last year that was, it was kind of a really interesting and well, I guess I called it a game changer for PWS genetics. He found 4 individuals who had a loss of function of one of the genes in the PWS region, the MAGEL2 gene, and those individuals had characteristics of PWS that included, all of them had autistic behaviors, all of them had intellectual disability, and to some degree or another they had obesity and hyperphagia, although that wasn't their major problems. The major problems was the autism and the intellectual disability. So, so that suggests that MAGEL2 may be one of all those genes that are in that area. That gene may be important in conferring those characteristics. So that's a real breakthrough as far as understanding the contribution of each of those genes to the PWS characteristics. So Dr. Schaaf happens to be at a place, Baylor College of Medicine, that has a really great resource, which is they do a lot of studies on autism and autism animal models, both rat and mouse models. So who knew, right? There are actually a battery of mice that have had genetic changes, genetically engineered to mimic autism. And those mice are very useful because they're a way to test treatments. So, so the MAGEL2 mouse, deletion mouse, already exists. Dr. Weverick, actually Dr. Weverick and Dr. Muscatelli each made different ones a few years back. They haven't really been studied extensively to see whether they have altered social behavior. So believe it or not, scientists sit around and try to figure out if mice social interaction is disrupted, and they have specific tests that they use for that. And as I said, the reason you would do that as a scientist is because then if you can characterize that and you can quantify that, then you can test interventions, you can test drugs, and you can see if that improves the behavior. And that's important because autism is caused by a lot of different disruptions in the circuitry of the neurons, and we're not exactly sure which flavor of autism PWS is. So we wouldn't, as therapies, as drugs are developed for autism, we don't want to just try each one in kids. We'd like to have some sort of screen to know which ones are going to be most important to our kids. So Dr. Schaaf is going to conduct a battery of tests on these mice and see if they can find a reproducible outcome that they can then use to develop new therapies. So as I said, this is important as a preclinical way to build up to a therapeutic intervention. And Dr. Schaaf is going to be attending the FPWR meeting this fall in New York, so he will hopefully be able to, or I know he will be able to, update us on his work then. And I think this is important work towards understanding intellectual disability and autism and PWS and thinking of ways to treat those features. The second study in this group of behavior is a study from Françoise Muscatelli. She has also made MAGEL2 deficient mice, and she's shown that these mice, in her hands, they do very poorly at feeding. So they really mimic that time right after birth that our kids, just are not good at feeding. But what she's also found is that if she gives them oxytocin right after they're born, they recover from that. And that's the basis of the study that Dr. Taubert is doing in France, giving oxytocin to newborn infants with PWS. So it's this, sometimes we wonder, like, why are we studying these mice with PWS? It does give you the basis for doing studies in humans and doing them thoughtfully and as efficiently as possible. So she has shown this in the MAGEL2 mice, and this study is really to get at the heart of that, to understand exactly what's going on and understand how best to translate that into humans. So is there a critical period in which they have to receive that oxytocin to have the effects? What are the long-term effects? How does the loss of MAGEL2 really affect the entire oxytocin system? It's a detailed look at those aspects of the oxytocin system and how MAGEL2 deficiency affects that. So we're excited about that because it connects the dots from a loss of function at the genetic level to something that we see in our kids. And the studies may help guide the development of new therapeutics, or, well, oxytocin is not a new therapeutic, but just help us understand how best to use that therapeutic approach. It will help us interpret the results of the ongoing clinical trials in oxytocin. So we're excited to fund this. We're funding this in collaboration with Prader-Willi France, who has been a great partner, has held lots of One Small Step walks, and those funds will go to Dr. Muscatelli to do this important study. Another study that we're going to be doing that has to deal with behavior is this study by Dr. Woodcock, who is actually just getting her first faculty position at Queen's University in Ireland. And she has, so she is championing a different way of thinking about PWS temper tantrums. She's been studying them and has really put forward this idea that the major trigger of a temper tantrum is task switching. So making our kids go from one thing to another. And I know many of you will be able to relate to this, that when your child is doing one thing and then now you want to make them do something else, they have a very hard time switching. And she's shown that in brain imaging studies, how they're not able to do that as effectively as typical individuals. And it's really interesting, her way of thinking. We all are told at some point to get your child into a routine because they do really well in routines, and that's very true. But what her studies suggest is, yes, they do very well in routines, but when you go to break them out of a routine, if something comes up and there's something unexpected, as we all know, that's a huge trigger. So she actually is advocating potentially, or looking at, throwing them a few curveballs so that they learn to deal with changes in routine. And she wants to develop a software training tool to help them learn how to cope with that task switching. So this is a really innovative and different way, I think, of thinking about our kids and how to help our kids learn to cope with actual life. And so they're going to be developing this intervention that is a computer-based intervention, and then seeing whether that can translate into real life, if it has an effect on people's lives. So this has the potential to have a pretty immediate impact on quality of life for everyone who lives with someone with PWS, and for the person with PWS. It's so frustrating for them to have a change in routine. If they can learn to cope better with that, that would be great. And so the potential outcome is an at-home software system that will help improve their task switching skills. The final grant in the area of mental health is a second year of support for Dr. Håkan Koegel, and I'm sure I didn't pronounce that right, so I'm sorry, in the Netherlands. And they have a very long-term study that they're doing on individuals with PWS in the Netherlands, looking at psychiatric problems, who's susceptible to psychiatric illness. And really the purpose of this study overall is to try to be able to identify who's at highest risk and then intervene before there's a serious problem. So we all know our kids are at a higher risk of having mental health problems. It's one of the most difficult things to deal with, I think. But if we were able to detect when those problems were going to happen and prevent them or decrease the severity, that would be huge for our families. So they're using a lot of cutting-edge technology, including this figure here showing diffusion tensor imaging. And this is actually showing the neural circuits in a human, a live human, so it's false colored, but that's the kind of imaging that can be done where you can trace neural circuitry. So they are looking at brain imaging in individuals with PWS by age. They're looking through puberty, which tends to be very difficult. Well, it's difficult for anybody, but tends to be particularly difficult for kids with PWS. And what they want to find out is can they detect an impending psychiatric crisis. And we're excited about this grant because it's proactive. We want to be able to predict who's going to have problems and intervene. And this idea is really gaining some traction in other areas of mental illness, that you can actually detect changes before the problem happens. And it's a really well-characterized set of patients. It's part of a longer-term, ongoing psychological and psychiatric project, so it'll have a longer-term reach. So this category, I kind of put two grants together. I don't know how well they go, but that was looking at the issue of how changes in the PWS region of chromosome 15 cause the overall changes that are associated with PWS, and are there some potential therapies to address some of the other issues in PWS. There's two studies in this area, one by Sue Kim, who was at University of Florida, has recently moved to Seattle Children's. And she has found, quite surprisingly, that we know that there's a change on chromosome 15 in the PWS region, but when she looks at the entire genome, so all of the chromosomes, she sees that there's changes across the genome in PWS. So she's just done this in a few patients, but this will allow her to study that. And it's hard to know exactly what that means. But it's definitely something that has broad implications for how these problems in PWS arise. And so she's going to be looking at SIB pairs and looking at this global methylation of PWS. There was just a study published on Down syndrome that showing again that even though Down syndrome we know is just an extra chromosome 21, somehow that change in the DNA affects all the other chromosomes, and that may be contributing to the very broad features that we see in Down syndrome. So wondering if there's something else like that going on in PWS. So it explores how the PWS region is impacting expression of genes on other chromosomes, and it's using some really good technology to look at that. And it might, in the end, identify new targets for therapeutic development that are not sort of the PWS genes. Another a project that we're really interested in is this a project by John Kremers in Belgium. And so this group is a very interesting story, and this is one reason that we definitely want to keep our investigator-initiated grant program, because nobody, I think people working on PWS wouldn't really have thought of this. So they study a really rare disease. PWS is rare, but this is even rarer, this hypotonia cystinuria syndrome. And that syndrome has, it's got some significant differences from PWS, but it has some similarities, including the fact that the kids are born very hypotonic and eventually go on to have obesity. So I'll just cut to the chase. These investigators have found that an existing drug can actually improve hypotonia in their patients with this hypotonia cystinuria syndrome. And this HCS is caused by a loss of a particular protein, or the part that causes hypotonia, this purple protein. So they study this disease, and they noticed that it looked a lot like PWS. So they said, hey, let's see if purple is also deficient in PWS. And sure enough, it looked like it was. And so what they're going to do is expand that study to really look at the deficiency of this protein in individuals with PWS. This is a protein that helps mediate the connection between nerves and muscle cells. They're going to confirm the impact on that neuromuscular transmission in a mouse model of PWS, and then they're going to explore whether the drug that seems to be helping their kids with HCS might also help kids with PWS. We like this study because hypotonia is really understudied. I mean, all of our kids deal with it early on, and we just kind of wait for it to go away or get reduced. This project incorporates both animal studies, so mechanistic, trying to understand what's going on, as well as a human clinical trial. So there's a therapeutic arm of this project. Again, this is an investigator who is new to PWS and is collaborating with an established PWS researcher, which is a great arrangement. And so the findings could be important for understanding the biological basis of hypotonia and PWS, and that's important, but perhaps more important to us in the short term is that it may offer a new therapeutic to help our kids become stronger earlier. The final two projects that I'm going to talk about are again looking at this concept of reactivating the maternal chromosome as a potential therapeutic for PWS. So the caveat here is we're a long way off from actually applying this therapeutically, but we'll never know until we try is kind of the thinking, number one. And the other thinking is all of these studies are going to help us understand the regulation of this region much better as well. So there's two projects that are going to look at this. One is from UC Davis, and that is the project by David Siegel. And this is the second year of a project. And Dr. Siegel, he is one of those people, I think, that is really interested in technology. So he's always, his career, he's always at the cutting edge of the development of new technologies. And one of the newer technologies are these artificial, so you kind of build them in the lab, proteins that can specifically activate certain genetic regions. So you stitch these things together using genetic tricks, and then you can put them into the cell. The one part of the protein will bind to a specific region of DNA, and then it will activate expression of a gene. So he has used this actually in Angelman syndrome, which you may remember is the sister syndrome of PWS. So it's loss of expression of the maternally expressed genes in the PWS Angelman syndrome region. So he's very familiar with this region of DNA, and he's been successful, at least in cells, of showing that he can activate gene expression. And so what he's doing now is essentially just flipping the technology a bit to try to activate the PWS region genes. So he is looking, he's going to be, this is the second year of his project. He's made the construct. He's really going to show whether it is functioning and what the effect is now. So what is the effect in a PWS mouse model. And he's doing it to activate the entire region, so all of the genes in the PWS region, and he's also going to make a construct that activates just MAGL2. The question being, if you could just activate that one gene, would you have a therapeutic effect? So he's going to do that and look at it in the mouse model of PWS. Sort of on another, it's not congruent with the rest of his project, but it just turned out that he was also making a rat model of Angelman syndrome. And in talking to him, it's actually quite easy to adapt that model to be a PWS model. So if the rat is bred through the maternal line, it's going to have little rats that have Angelman syndrome. If it's bred through the paternal line, you'll get little rats that have Prader-Willi syndrome. So why are we interested in a rat with Prader-Willi syndrome? And that's actually because the autism community has shown that these are very good models for autism, for doing cognitive studies, and for evaluating drugs that can improve cognition and improve autistic qualities. So we're excited about this grant. It's going to do two things. One, it's going to establish whether gene therapy is feasible and whether it has any beneficial effect in PWS. The advantage of this approach as opposed to some other approaches is there's a high degree of specificity. So it should just activate PWS region genes and not other genes. The group has already shown proof of concept in an Angelman mouse, and they're also going to be developing this new animal model that will be a resource for future work. So this is exciting because, as I said, it's potentially a specific way to activate genes in the PWS region. The other approach, so there's many different approaches that we can take to try to turn these genes on, and I really think we're going to have to try out a few of them to see what makes the most sense and what can go forward into a potential therapy. So the other approach that we are planning on supporting is a second year of Dr. Zhang's study at Duke University. So as we've said, there's growing evidence that supports that reactivation of the maternal allele may be possible. We haven't shown whether it has a therapeutic effect, but at least it may be possible. So Dr. Zhang has developed an assay where he can screen lots and lots and lots of drugs at once in this high throughput format. So he takes PWS cells and he has an assay where if the PWS region genes get turned on, the cells turn green, so you can pick them out. And he is screening for drugs that specifically turn on PWS genes. And he's done that in a small library, and he's detected a couple of compounds that seem to be able to do that. And he's going to, in this part of the project, he's going to look at those compounds more fully. The other thing is he's going to do is screen an additional 10,000 compound library and try to detect, so not every compound that you pull out is going to be suitable for developing into a drug that people can take. So you need a couple of candidate compounds. So he's going to gain some additional compounds, screening a second library of compounds. So we're excited about this because small molecules, so it's much easier to give an oral drug than it is to give a gene therapy. So from that perspective, this approach has some advantages. It's the first step towards an oral drug that potentially would be able to treat the underlying problem. The group has a lead compound from the first screen, so they've shown that they can do the screening. This is an individual who has worked in PWS before. He works in Angelman syndrome. He's kind of committed for the long term in PWS. And he's laying the groundwork for new drugs that might treat PWS at the genetic level. And as I said, we had set aside $750,000 for these grants. We couldn't resist going over a little bit because we were so excited about the grants. And to kind of make up the difference, we've started a puzzle project in which we're giving our donors an opportunity to buy pieces to the puzzle in support of Dr. Zhang's project. So that is hopefully a way to get additional funding so that we can move that project along quickly. So that is all of the projects. What did we leave on the table? So our goal is has always been to, so we rate the projects. We have the scientists and the advocate reviewers rate the projects from all the way from exceptional and outstanding to excellent down to very good and good. Our goal is any project that is rated as excellent or better by both the scientists and the advocate reviewers should get funded. That's what our goal is each year. And sometimes we're able to do that and sometimes we're not. I have to tell you, it just it kills me to send a letter or call up a scientist whose project has been rated as excellent by both the scientists and the advocate reviewer and tell them that, you know, we're not able to fund it even though everybody thinks it's an excellent project. So these are projects, we've put them in the fund if category, and we're going to hope that our fundraising continues as strongly as it has been going. And if we reassess our financial position and are able to fund those will be the next 3 to fund. So we'll keep you attuned to that and let you know. I think they're exciting projects as well. So I hope we're able to do that. So that's the end of our projects for this year. What's on the horizon for us? As I hope everybody got the first newsletter, the clinical trials alert, which showed several clinical trials that are just starting. If you didn't get that, you can email me or Susan and get on the list to get that. So we'll be updating that on a monthly basis to let you know. As we've said before, there's a lot of clinical trials that are coming down for PWS. We're really excited about them. It gives us a lot of opportunities, but you know, we have to these trials aren't going to get done if people don't make a commitment, and it really is. It's a significant commitment to learn about the clinical studies and potentially enroll in the trial. So we want to make that as easy for you to learn about those trials as possible. To that end, too, we're currently working with 2 investigators as part of our clinical trials initiative to provide funding, hopefully in the fairly short term, for 2 clinical trials in PWS. So we're very excited about that, and we will let you know. Right now we're working with those investigators to bring the projects to the point that they're approved for funding. And we're engaging, and you'll hear more about this in the coming months. I think this is a critical thing that our community needs to do, is engage the Food and Drug Administration and we have a plan that we're going to roll out and ask for comments on to give the FDA feedback. So all these clinical trials are going to go through these studies, and then the FDA will have to decide whether to approve them or not approve them. So they need to have a good understanding from our perspective, the patient perspective, of what's important to us, what sort of risks and benefits, what's that risk-benefit ratio that we're willing to tolerate is. So we want to be able to provide a voice to the FDA as they make those decisions. So that's the clinical trial side. We're also looking the next couple of months. We're starting to work with Rare Science, and which is a nonprofit research organization that brings together resources to help develop new therapies for rare diseases. And so we're really excited about that collaboration. And finally, we are getting ready to launch the global PWS registry. I appreciate everybody's patience on that, and we'll be launching that soon. I think that's probably what our next webinar will be about. So we're excited about all of those things. A lot of great things happening on the horizon. So I just wanted to end, and I know Susan will have your questions, but I wanted to end again by thanking all of our supporters, all of the people who work so hard. I know you guys have so much on your plate and are willing to raise funds for PWS or contribute in so many other ways as well. So really appreciate that. So Susan, do we have any questions? that
We
have
sure
popped up?
do. So, first of all, Teresa, thank you very much for disseminating this information so that our supporters can understand it. 10 projects is a lot to try to take in, and I think you did a great job presenting those to us. So, we have a few questions here. The first one is, what percent goes to expenses and admin fees? This is a question from James Loker. James, in 2013, 82% of our expenditures went directly to research programs, 4% went to administration, and 14% went to fundraising. If anyone has further questions regarding our finances, they are available on fpwr.org. If you look under our foundation, there is a page dedicated to our financials, and you can download not only our annual report, but also our 990s there. The next question from Gloria Wolshine from Canada. Teresa, you showed us a list of over 50 studies that were published. Where can we find more information about those publications?
So those are also available on the FPWR website. So Jessica has gone through and linked publications to each project. So I think if you click on a project, and it should also have the publications that are associated with that. We also have a page that is just called Research Outcomes, and that includes the publications as well as some of the other outcomes, such as resources that have been made available, new funding that investigators have gotten. So please look through that, and we try to keep that up to date. It's a little, I mean, it's a great problem to have that, new papers are coming out all the time. The investigators are all supposed to, give us a copy as soon as it comes out, and sometimes it takes a little while. So we do our best to keep those up to date. But yeah, please look on our new website, which allows us to, which I think is a great advantage now, to have the project and then have the outcomes that have come out of the project, so you can see, what has happened with those projects.
Mm-hmm. Wonderful. Our next question is from Rebecca McWilliams. What would be the next step if Dr. Jang were to discover a candidate compound?
So the next step, so this is very early. I had slides, perhaps you've seen before, my slides on the drug development process. So this is very much on the left-hand side of it. It's the first step. So the next thing, now he's screening libraries that are known to have some properties that are good for drug development, but you would still need to see, so any drug that he pulls out is going to have to cross the blood-brain barrier, because we're talking about activating gene expression in the brain. That's probably the most important place. So you would have to see that it gets into the brain. You would have to look at toxicity to make sure it doesn't have any off-target effects. So other things that you wouldn't expect. You have to make sure that it would be specifically activating the PWS region and not 1,000 other genes. So the next step in that process is to do those studies, but also do potentially medicinal chemistry. So take the compound and then modify it, and then kind of go back and see, does that reduce, if there's some toxicity, does it reduce the toxicity? So medicinal chemists do this iterative process where they test the compound, they figure out why it's not exactly right, they modify the compound, and then they test it again. So they would do that, and then they would do the preclinical studies, which is looking at it in animals and making sure, as I said, that it does what you think it does and it's not doing bad things. And then it would go into humans. So it's a long process, usually on the order of 5 to 10 years to get it to the point. And that's why he needs several compounds, because most of them will fall off along the way.
That's very interesting. Our next question is from Christy Polkerbeck. Do we have any information to share on when the next Bulletin trials are taking place, and where can one go to find more information?
Well, so our clinical trials bulletin that we're sending out every month will have information as soon as the company announces that they'll be doing a clinical trial. I think it's fair to say well, I know it's fair to say because it's public information, that they're planning on doing a trial in 2014, in the latter part of 2014. So that would be a phase 3 trial, and so it'll be coming soon, and we're excited about that, and we will let you know as soon as they've finalized it.
All right. I don't have any additional questions in the queue. If anyone does have a question, please feel free to type it out. We'd be happy to answer them. We could give everybody perhaps another minute. Looks like we're right on time. Teresa, we'll be wrapping up right at 6 o'clock Pacific, 9 o'clock Eastern.
Right over the dinner hour.
Exactly. All right. Here's a question from Bob Barnard. Are there any natural sources of oxytocin?
Hugs. I mean, seriously. So I mean, I'm not being too facetious in that. So when we have human-human interaction, or I guess human-dog interaction as well, when we have positive social interactions, we do release oxytocin, and so if you know, with your child with PWS, the more that you can